Hello friends. Today we are going to discuss about neurobiology of treatment-resistant depression. I am Dr. Suresh Badadmat, Professor of Psychiatry, working at National Institute of Mental Health and Neurosciences, Bangalore. In this video, I'll be specifically discussing about molecular mechanism, what is the genetical mechanism, neuroimaging studies, basically the neurobiology of treatment-resistant depression. This video is very important. If you understand the neurobiology behind the treatment-resistant depression, the researchers can focus on how we can crack this treatment resistant depression. As you know, 60% of the patient do not show improvement after the first antidepressant trial. That means once a patient is diagnosed as depression and if you start medication, 60% of them do not respond. Subsequently, even after 4 trials of antidepressant, 30% of them do not show improvement. Further, only 30% of them remit completely with the depression. Remaining of them either will have partial improvement or treatment resistance. The treatment resistance has a debilitating consequences. It causes increased morbidity and mortality in the form of suicide. Morbidity in the form of loss of job, unemployment, substance use. They would like to treat their depression with alcohol, drugs. They may not go to the job. Family discord, marital discord, and also difficulty in upbringing their kids. And this translates into heavy cost, not only on the patient, family, and also on the community. There are many studies have clearly said that the cost on the nation is huge because of depression. Now let's move into the neurobiology of treatment-resistant depression. Here, we need to find certain markers which will indicate, yes, this patient has treatment-resistant depression. How to know that? So there are various approaches have been taken. One is candidate markers. Here, they look into the clinical indicators, neuroimaging indicators, genetics, epigenetics, proteomics, metabolic, and biopsy. So these are the various studies have been undertaken. Once the candidate marker have been studied, invariably it talks about whether it predisposes a patient to a depression, whether this marker can be used for diagnosis, can we stage the treatment response, can we predict the outcome, or else can we predict the relapse. So these biomarkers can be applied, whether we can do early identification, can we manage diagnosis, can we subtypes various classification of depression, can we know how the disease progress, treatment response, and also whether the person is high risk for suicide. So these are the various ways by looking at the biological markers we can understand depression. First and the foremost hypothesis with regard to treatment resistant depression is neurohormonal theories. As you know, serotonin, epinephrine, dopamine have been suggested with regard to onset of depression. Invariably, dysfunction in serotonin, norepinephrine, and dopamine pathway has been considered for depression. Invariably, if there is any dysfunction, either in the production, response, receptor, or else in metabolism of these neurohormones can cause treatment-resistant depression. Further, now the glutamate and GABA has been implicated in terms of treatment-resistant depression. There are some studies from the imaging, especially from MRI studies and functional MRI studies, and also magnetic spectroscopy has also been considered. Chronic stress increases cortisol level in the blood. This cortisol level causes impaired neuroplasticity in the hippocampus. Hence, certain areas of the brain, including hippocampus volume, if it is decreased, that is cortical thickening or thinning. If the volume decreases, those are the patient may not respond to treatment resistant depression. There are various MRI studies which clearly indicated those patients who do not respond to treatment have found that their cortical hypovolume has been found. This image clearly indicates about the low baseline volume of hippocampus. Further, other studies have shown gray matter in various cortical and subcortical areas. Thinning has occurred, and these patients do not respond to treatment. Why this cortical thinning occurs? It is still in the investigation. One of the mechanisms as suggested is neuronal plasticity decreases. Hence, those medications which we give may not enhance either the neurohormonal plasticity or receptor plasticity or neurochemical plasticity. Hence, the response becomes less. One of the area, especially in the brain, anterior cingulate cortex has been suggested. This anterior cingulate cortex is a critical brain region involved in emotional regulation, decision making, and cognitive control. What is this cognitive control? Is rumination. Especially in OCD, we can see obsessive rumination. Similarly, in depression, we see depressive rumination. Anterior cingulate cortex, especially in the cognitive control, that is, depressive rumination, worsens whenever there is a dysfunction is anterior cingulate cortex. Various MRI study and EEG studies have found that high theta activity in the anterior cingulate cortex reflects cognitive emotional dysfunction, excessive rumination, impaired control on emotion. That means the studies which have found in imaging and also EEG has been found to be positive. And you can say elevated baseline activity, that is theta activity in anterior cingulate cortex has been implicated. Further, translocator protein has been implicated. This translocator protein is a vital protein which is found in the mitochondrial structure, and it is specially present on the outer mitochondrial membrane. And this is expressed in microglia of the brain. It is implicated in several cellular functions, including steroidogenesis, mitochondrial energy metabolism, and immune regulation. This translocator protein being in the mitochondria, of course, it is involved in energy and also steroidogenesis and immune regulation. On PET imaging, it has been shown increased translocator protein expression in the brain region, especially in the prefrontal cortex, anterior cingulate cortex, and hippocampus. By increased translocator protein, that means there is involvement of this translocator protein, and it activated microglia produces pro-inflammatory cytokines such as interleukin 6, tumor necrosis factor, and interleukin. By increase in these neuroinflammatory products has direct effect on the brain and subsequently in depression. And if this activated microglia, if we do not treat them properly or it becomes treatment resistance, the pro-inflammatory cytokines have been found to be elevated in various studies. Now let's move into the neuronal connectivity. Again, studies have implicated that there may be, especially in treatment resistance, they found that either the hyperconnectivity or disconnectivity. Hyperconnectivity in certain region can cause treatment resistance, or else disconnectivity can cause also. Although these results are inconsistent across study, some studies have found that yes, hyperconnectivity in certain region causes treatment resistance and disconnectivity also causes. You need to understand the triple network dysfunction model. The triple network are those networks in the brain which are very essential to understand the treatment response in neuromodulation or else in ECT. There are three important neural networks has been considered. One is salience network or central executive network, and it is often also called as cognitive control network. Some authors consider both are almost similar. Some authors says that no, it is different. But I believe both central executive network and cognitive control network are same. And finally, the default mode network. Let's understand salience network. This is the network which controls both default mode network and also cognitive control network. This salience network consists of insular cortex, dorsal anterior cingulate cortex, amygdala, and temporal pole. And it is involved in interoceptive awareness, task set maintenance, and detection of salient stimuli from the environment. Although these are the cognitive work, but this is the one which monitors both default mode network and cognitive control network. Now let's understand about cognitive control network. Here it starts from dorsoprefrontal cortex and also dorsal medial prefrontal cortex. From there, dorsal anterior cingulate cortex, posterior parietal cortex, inferior temporal gyrus, and also precentral gyrus. And it is involved in attention demanded cognitive tasks and also top-down modulation of working memory and attention. That means here it plays a very crucial role on demand involvement of attention. As you know, especially in depression, the attention and concentration decreases, and of course, they also report of decreased memory, that is working memory. Now let's understand about default mode network. This is the network which involves prefrontal medial cortex and it projects towards the posterior cingulate cortex and precuneus cortex, lateral temporal cortex, temporal lobe and pole, hippocampus is involved, and this network is involved in emotional processing, self-referential mental activity, mind wandering, recollection of experience, and attentional demand tasks. This default mode network, when you are not actively involved, or else you are doing certain activities like driving, moving around, in such a scenario, the brain will be in constant activity. It will be thinking something that is called as default mode network. So these three triple mode network which I am talking about plays a crucial role. Along with this, there is another sensory motor network. This is involvement of thalamus, putamen, supplementary motor area, primary motor cortex, somatosensory cortex, and insular cortex. This basically involves in exteroceptive awareness and motor activity. So these four networks play a crucial role in understanding about treatment resistance. This diagram clearly indicates that the salience network plays a crucial role in switching between default mode network and cortical control network or cortical executive network. If there is any imbalance between this, either there may be treatment resistant depression and switching does not occur at time, the patient may switch between depression and mania. This has been proposed. However, some of the studies support this network hypothesis and many of the studies do not. But however, this is the recent network hypothesis has been proposed. Further, if you look at the hyperconnectivity within the default mode network, particularly in the medial prefrontal cortex and posterior cingulate cortex, results in increased rumination and further treatment resistance. Decreased connectivity in the cognitive control network, or we can call it as cognitive executive network, and here this affects the goal-directed activity and executive function, especially in depression. And if you give treatment, especially with transcranial magnetic stimulation and ECT, some of this can be reversed. Although this has been proposed in depression, many a time these networks dysfunction. How it occurs is still a matter of debate. Further research is needed. But however, treatment resistance do occur in these network areas. And this diagram clearly indicates the default mode network, which is in the green, central executive network, which is in the yellow, and cognitive control network, which is in the orange. You can see if there is a decreased activity, which is in the blue, and increased activity, which is in the red. In certain area, especially in default mode network, if there is hyperactivity and decreased activity in central executive network can cause treatment resistance in the long run. Now let's look into one of the important publication by Runai and his colleagues in 2022. This study was published in Neuroscience and Biobehavioral Reviews. The title of the study was The Neurobiology of Treatment-Resistant Depression, a systematic review of neuroimaging studies. Here, they looked into various studies and did the review. What are the findings in treatment resistance? If you look at the summary of this study, there is reduced functional connectivity within the default mode network. Although there is hyperactivity, which is studied in the previous study with EEG, they found that reduced functional connectivity, that means which is functionally should be there, it is not occurring, but there is hyperactivity and depressive rumination. Rumination does not cause any work, hence there is a functional connectivity is reduced. Reduced functional connectivity between the components of default mode network and other brain regions also plays a crucial role. Further, hyperactivity and default mode network also plays a crucial role. These are the findings of this study. In addition, aberrant activity and functional cortex in the occipital lobe may play a role in the treatment-resistant depression. So these are the findings of the neuroimaging studies. This can be clearly depicted. The outcome, if there is red color, the outcome is very poor. The greens suggest more activity. If you look at the 5-HT1A, serotonin, and MAO of the enzymes or transporters. So this has been implicated and studied in various scientific research labs. Now let's look into one of the landmark article which was published recently. Kujumba and his colleagues published an article in Molecular Biomedicine. This is a landmark article. If you want to know more about treatment resistance, please go through this article. This is Treatment Resistant Depression, Molecular Mechanism and Management. This study looked into comprehensively with regard to treatment resistance and also suggested management. First and the foremost, they talked about monoamino neurotransmitter interaction in depression and treatment response. You can see this diagram. This diagram clearly talks about 4 important neurohormones. One is dopamine, serotonin, and norepinephrine. So, dopamine deficiency causes somatic domain presentation in depression, characterized by motivational deficit, psychomotor retardation, emotional responsivity, and other depressive symptoms. That is dopamine deficiency. If there is norepinephrine deficiency, affective cognitive domain, characterized by feeling sad, irritability, and cognitive symptoms. If the dopamine deficiency is being implicated, they are involved in treatment resistance. Here, this clearly indicates that if there are involvement of inflammatory cytokines, especially tissue necrotic factor, IL-6 and IL-1 beta, so the interleukins, this can directly affect the metabolism of dopamine, serotonin, and norepinephrine. They will be sent back to the neurons immediately, and non-availability within the synaptic cleft crosses the treatment resistance. So this treatment resistance, again explained by monoamino neurohormones, has been implicated. Now let's look into the dopamine as a neuroinflammatory moderator in depression. You can see in this diagram, there is a blood vessel. That means if there is any neuroinflammation occurs and there the blood-brain barrier decreases. In this, there will be release of pro-neuroinflammatory cytokines, TNF, IL-10 and IL-13. This enters into the brain. From there, it causes decrease in dopamine. That is, these pro-inflammatory cytokines acts directly on the presynaptic neuron, and the uptake of all these dopamine occurs, and there is little availability of dopamine. And this results in either microglia will be activated into M1 phenotype, and further pro-inflammatory cytokines are released, and it goes into a cycle. More the pro-inflammatory, less the dopamine. Less the dopamine means microglia gets converted into M1 phenotype of microglia and further increases pro-inflammatory cytokines. If by treatment, if the dopamine start releasing, and then it causes M2 phenotype of microglia, this reduces pro-inflammatory cytokines but increases anti-inflammatory cytokines. That means inflammation decreases. Here, tissue necrotic factor beta will be released, interleukin 10 and 13 will be released. This is very essential in response with regard to depression. So this has been considered in treatment-resistant depression. Please understand, neuroinflammation has become one of the important mechanism of treatment resistance, treatment response. Now let's look into the pro-inflammatory cytokines induced kynurenine and tetrahydrobiopterin enzymatic pathway. Again, you can see there are two important pathways. One is BH4 pathway. It is also called as tetrahydrobiopterin and kynurenine pathway. In this pathway, you can see there are involvement of cytokines, and these pro-inflammatory cytokines will lead to formation of further either quinolinic acid or kynurenic acid. If it is a dopamine neuron, then quinolinic acid will be involved. That is, the first one is tryptophan. If the tryptophan has to be converted into 5-HTP and serotonin, then there is, because of the inflammation, indoleamine 2,3-dioxygenase, there will be inhibition. This lead to kynurenine involvement. From the kynurenine, it will lead to quinolinic acid, which causes treatment-resistant symptoms. If the tryptophan gets into serotonin formation and formation of kynurenic acid, this causes increased BDNF, that is brain-derived neurotropic factors. This BDNF is an anti-inflammatory and also neurogenesis, and treatment responsive symptoms occurs. Hence, my dear friend, the involvement with regard to the inhibition of certain enzymes and also the availability of tryptophan, and leading to which one? The quinolinic acid increases, which leads to decreased BDNF. If there is kynurenic acid occurs, increased BDNF occurs. Invariably, it can be considered as the neuroplasticity and the protein formation. This has been implicated. Now let's look into the stress HPA axis. As you know, the stress causes increased cortisol. This cortisol will bind to the glucocorticoid receptors in the microglia, especially in the brain. And this results in either increase in the mRNA related to cytokines, that means pro-inflammatory cytokines will be released. Further, increased extracellular ATP, which further increases the astrocytes or stimulation of astrocytes then into microglia, and more of pro-inflammatory cytokines are produced. As you know, more the pro-inflammatory cytokines, these cytokines decreases either the dopamine, serotonin, or norepinephrine in the synapses, either by the receptor mechanism or else increased reuptake. And if there is a dysfunction at this level, the treatment response becomes very poor. Now let's understand about the astrocyte dysfunction that is mediated again by the pro-inflammatory cytokines. If you look at the astrocytes which are there, and this astrocytes dysfunction can occur because of the cytokines interleukin 1 beta, interleukin 2 and 6, and even 12 has been implicated. These interleukins can cause dysfunction of the astrocyte, and this will lead to glutamate increased production, and further the glutamate uptake will be reduced. Hence the glutamate is available in the synapses, hence there will be hyperstimulation. This results in hyperglutamatergic state, and similarly in the GABAergic neuron there will be depletion of GABA, hence there is a dysfunction with regard to imbalance between the glutamatergic and GABA. As you know, this dysfunction can lead to the treatment resistant depression, and invariably the GABA depletion, which results in direct action on the dopamine neuron, and this dopamine neuron will result in the treatment resistant depression. Here there will be more of biological symptoms, especially retardation, depressive symptoms. This is very difficult to treat. Now let's look into the genetics of treatment resistant depression. Depression being a heterogeneous disorder, polygenic trait, there are many genes involved, there are phenocopies. Some patients have a distress whenever there is a stress, and many a time long term dysthymia is there. Hence there is a variable presentation, variable genetic markers or genetic presentation, epigenetic phenomena, and heritability is around 20 to 50%. Environmental factors, childhood trauma, and variation in phenotypes becomes very difficult to understand the genetics. However, I'll be discussing those studies which have been replicated. The genetic mechanisms of treatment-resistant depressions are very complex. They are multifactorial in nature. The key factors include genetic variation, especially in the neurotransmitter system. Those have been studied, that is serotonin, dopamine, and glutamate. There are genes related to neuronal plasticity, especially BDNF has been implicated. Immune system regulation and dysregulation studies are pouring in, especially the pro-inflammatory cytokines have been implicated. Further, the drug metabolisms, that is CYP 450 enzymes, either the fast metabolizer, if the fast metabolizer is there, that means the medicine is not available for action. So these are the studies have been done since past 3 decades. Some promising findings with regard to decreased BDNF levels, that is BDNF Val66 MET gene polymorphism is associated with resistance to SSRIs. That means, as I mentioned earlier, the BDNF level increases, that means there is a response. If there is a polymorphism and low level of BDNF or altered BDNF which is useless, if it is there, then there will be poor response to treatment. Further, antidepressant resistance is associated with polymorphism in SLC6A4 gene in 5-HT2AR receptor, and this is a serotonin transporter gene. Again, this may cause dysfunction in the transporter gene, and that will result in a transporter receptor is malfunction or dysfunctional. Hence, there will be a treatment resistance. There are some promising genetic findings, especially with regard to NMDA receptor subunits. This A variation, particularly in GR1 N2A in a gene which encodes NMDA receptor subunit 2A, has been implicated with regard to treatment resistance. Similarly, with regard to glutamatergic neurotransmitter system, increased risk of treatment resistance is associated with RS 12800-734 and RS 195-4787 gene. And this variant is included with the GR1K4 gene, especially encodes for kainate receptor and NMDA subunit receptors. These are the genetic findings which have been found promising. Further, a study by a European group of study of the resistant depression group involving 2,762 patients in 8 European countries found single nucleotide polymorphism, that is SNPs, within the GAP 443, ITGB3, triple P3CC, and ST8S1A2 and CHL1 gene to be linked to treatment-resistant depression. This being one of the largest population study across 8 countries, has been considered one of the promising. Epigenetic mechanism such as DNA methylation, histone modification, and non-coding RNAs have been found to influence the gene expression or neuronal plasticity without altering the underlying genetic code. There are some inconsistent findings across studies. That is, cytochrome 450 enzyme is also been considered as responsible for treatment resistance. There is variation in the gene encoding with regard to enzyme CYP2D6 or else 2CA19 has been lead to altered drug metabolism has been considered, that is fast metabolizer and slow metabolizers. This has been implicated, but again, the studies have been found to be inconsistent. Another very important, again, which is inconsistent finding, and this is in the early part of research, that is mechanistic target of rapamycin. Pathway is involved in synaptic plasticity, protein synthesis, and cell growth. If this mechanistic target of rapamycin pathway is get altered, then there is a possibility the neuronal plasticity may not occur, or else there may be dysfunctioned neuronal plasticity. That means neuronal plasticity with regard to increased neurochemical production, transporter receptor itself, and various other genetic modification which has to be neuronal plasticity. If there is a dysfunction, may impair the neuronal plasticity, and of course, antidepressant may not work in such patients. Further, COMT genes have been implicated, especially Val158 MET polymorphism has been implicated, but again, this has been found to be in the prefrontal cortex. Again, the studies are not consistent. The other interesting study is human endogenous retrovirus. This is an ancient evolutionary retrovirus RNA which is there in 8% of the modern humans. DNA are typically epigenetically repressed but can be reactivated intrinsically or through extrinsic factors. These are those retrovirus which are present as evolutionary part. This gets activated either intrinsically or extrinsically. Extrinsically, you can consider as a stress. That stress stimuli may activate the microglia, and those can produce pro-inflammatory cytokines, or else these microglia themselves can increase these retrovirus, and that can produce more mRNA, further increases pro-inflammatory cytokines, and that results in depression resistance. To summarize, my dear friends, due to the heterogeneous nature of depression, various phenocopies, different types of depression, presence of stress-related distress, and also polygenic involvement, epigenetic mechanism, this becomes very difficult to study the neurobiology. However, the review reveals some underlying mechanism. There is no any candidate biomarker, but the combination of biomarkers need to be studied. That means we need to look into various neurotransmitters, inflammatory mediators, stress, HPA axis dysregulation, genetics, epigenetics, and neurophysiological factors. That means one factor is not there. Since the depression has multiple pathways, hence there are multiple biotransmitters or biomarkers need to be studied. Again, the salience network, central executive control network, and default mode network. The salience network is the one which balances between the central executive network and default mode network. If there is any dysregulation in this network, can also lead to treatment resistance. To conclude, treatment resistance neurobiology involves an interplay between various factors, not a single factor. Hence, the candidate biomarker has failed. We need to take all the biomarkers, more the biomarkers, then there will be a stronger prediction of treatment resistance. The lesser the biomarkers, the poor is the prediction. Hence, we need to involve neurotransmitter system, inflammatory mediators, stress, HPA axis, genetics, and other neurophysiological factors has to be studied. Effective management of treatment resistance requires targeted several interaction mechanism to be studied with regard to various pharmacotherapies, neuromodulation, and of course, therapy has to be considered. Thank you very much for giving your valuable time. Stay safe.